Thank you, and good afternoon, everyone. Thank you very much for the invitation to speak. I wanted to tell you a little bit more about Medica and then what we're doing most recently. The company has been around for a while, set up as a T-cell immunotherapy company, and that's really been our focus since inception. We've accumulated over time three very exciting platforms based on T-cells, a non-modified viral-specific T-cell platform, a CAR-T/CAR-NKT platform, which is being developed in partnership with the Baylor College of Medicine in Houston, and the dominant TCR platform that was developed at UCL, University College London. And I want to focus a little bit more on the first, the two genetically modified platforms. Our investors are mostly UK-based funds, Woodford, Invesco Perpetuate, and Touchstone Innovations. Touchstones used to be known as Imperial Innovations. Some of you may know them in that name. All three of those are evergreen funds, so we're able to fundraise regardless of their funding themselves. We also have the Wellcome Trust and CIPRIT, the cancer funding from Texas, which is funding some of the work we're doing with Baylor in Houston. So this is our current pipeline, and you can see a lot of it is still in late preclinical stages. There are actually 5 programs, 3 with Baylor on the CAR-NKT platform and 2 with UCL on the dominant TCR platform that are destined to enter the clinic within the next 18 months. There's obviously quite a lot of work, but it certainly helps being able to work with partners and being able to share the workload with these partners. And certainly both of these are partners with a lot of experience in running clinical trials with genetically modified T cells. The first, and I'll talk more about the platform in terms of the dominant TCR and how it works. WT1 is a target that we in-licensed from the cell therapy catapult a few months ago in early summer. That's the first dominant TCR that we're going to be developing. I'm naming that specifically because it came to us with some ongoing work that the catapult is performing in terms of optimizing further the manufacturing process for the T cells that are modified with the TCR. And also it enables us considerably by giving us access to a GMP manufacturing facility that the cell therapy catapult have established in Stevenage, northwest of London. Survivin is the next one. That's a target that came to us through the Baylor collaboration, and it is now being worked at UCL to improve the performance, as I'll show you on how the dominant TCR platform works. So let's start first, and I won't go into too much detail due to time limitations, but I wanted to talk a little bit first about the CAR-NKT platform and explain to you why we feel that the natural killer T cells, which are a subpopulation of T cells in the body, may be a better cell type for CAR therapeutics. And that's based on their biology. First and foremost, these are invariable T cells. So their TCR is specific against pathogen antigens. It's a glycoprotein called alpha-galactoseramide, and that's found on a number of pathogens that infect us. And so the fact that these cells are invariant means that their TCR will not target any human antigens or any eukaryotic antigens at all. And therefore, these cells will not cause graft versus host disease, whereas other T cells, of course, will. And that's obviously very important for a number of reasons, chiefly among which obviously is the fact that we can develop off-the-shelf products using NKT cells without the need to gene edit the TCR out of the cells. And that obviously makes the process considerably simpler than a process that's based on T cells, whereas the TCR is removed by a form or other of gene editing. And that's obviously a considerable time-saving. But in addition to that, we all know that TCR is an important parameter in T cell biology. T cells use the TCR for being able to grow. They receive trophic signals through the CD3 that is constantly, obviously, on the TCR. And in addition to that, being able to expand the cells using the standard CD3, CD28 beads would not be possible when the TCR is missing. So not only are we giving ourselves a head start by not having to remove the TCR, but obviously we're generating cells that are easier to grow. In addition to that, it's very easy to generate GMP-grade alpha-galactoseramides, so we can grow these cells using alpha-galactoseramides rather than having to use beads like one needs to do with T cells. And that means that it's a much more physiological, much more natural activation, which leads to a T cell population that is much richer in central memory T cells, CD62 ligand positive T cells, compared to a T cell process that uses CD3, CD28 beads. And obviously, we're seeing a lot fewer cells expressing exhaustion antigens because these cells are growing in a more natural and a much better way compared to the beads. So these are two very important reasons as to why we chose NKTs instead of T cells. But in addition to that, there's also other biological factors implicit in their biology. They are known to work very well under hypoxia. They are known to home very well into solid tumors. These are cells that are meant to be found in tissues, in deep tissues, and so this is really where they're best tuned at. And we felt that. For Cell Medica, our strategy being targeting solid tumors rather than hematological malignancies, these cells are probably a better choice for targeting solid tumors. So our platform is based on these cells. In addition to using the NKT cell, we also have the addition of a cytokine secretion, a constitutive secretion of a cytokine by the cells. IL-15 is obviously a commonly used one, and when we've selected that as well for our products, there is enough proof of concept there to give us the confidence that this will certainly make some difference in solid tumors. And again, with the understanding that a solid tumor, an immunosuppressed environment, the cells need more than just a CAR in order to activate and remain activated. We also bought last year a company called Delanex Therapeutics in Switzerland. We're using this company to generate the single-chain fragments for the CARs. They have the ability to optimize them based on stability, based on their ability, or obviously the lack of tonic signaling, which is very important, as well as the fact that they're humanized and therefore will not cause an immune response against them. So that's all I wanted to say about the NKT platform. I want to move on to the dominant TCR, which is a new platform. It hasn't been published yet, but will be very shortly at UCL. And it really is based on the knowledge that TCRs in T cells will express at varying degrees. And the concept of a strong versus a weak TCR has been known for over a decade now. And then in addition to that, we have to contend with the fact that we are adding our TCRs into a T cell that already has an endogenous TCR. So the two TCRs will compete. Namely, the most important bottleneck is the amount of CD3 that's available in a T cell. A T cell will not express too many TCRs, so about 100,000 is an average for a T cell. So introducing one's TCR into a T cell and having it compete with the endogenous is bad enough. We also have, obviously, the limitations of transduction efficacy. So in many cases, a TCR-modified T cell is not going to express sufficient levels of the TCR in order to become activated against this antigen. And so based on that knowledge, what Hans Staus did at UCL is sequenced a huge amount, about close to 1,000 TCRs, either weak or strong, and lined up the sequences and came up with a list of amino acids, close to 30 amino acids, which are more often found in strong TCRs versus weak TCRs. And obviously the next very obvious experiment is try and see what would happen if you switch these amino acids over. So what I'll show you here is an experiment where a list of 14 amino acids known to be contributory to a strong TCR are switched around between a strong and a weak TCR. So what you can see here is a strong TCR that is inserted into a T cell line that doesn't have an endogenous TCR. And you can see that out of the positively transduced cells, about 90% of the cells will express that TCR. A weak TCR put into the same cell line will express at lower levels. Now, if you take the strong TCR and you exchange 14 amino acids known to be strong into weak amino acids, you almost completely obliterate the expression of that TCR. And on the other hand, take a weak TCR and exchange these amino acids for strong, and about 90% of the cells will express that TCR. So you've converted that TCR into a strong TCR. Moving into the more competitive situation, a more natural situation, where the jerked cells now are expressing another TCR. This is known to be a strong TCR. Immediately, we lose expression of the strong or so-called strong TCR. So about 60% of the T cells are expressing that TCR. The situation is a lot worse with that weak TCR because that strong TCR is really taking over in that cell. It's the dominant TCR. And so a very small amount of the T cells will express. And obviously, as I mentioned before, these are all the positively transduced cells. So if you look at it from the perspective of a 30 to 40% transduction efficacy, as you often see in the clinic, that's too small a number of T cells. These T cells will not activate. Again, take the strong TCR and convert it into weak. It becomes completely useless. The weak TCR, however, you can convert that now into a dominant TCR. And that's really the important parameter, and I'll show you why this is truly important. I also want to point out that what you're seeing here is percentage. If you're actually looking at the fluorescence intensity, that's about threefold higher. So the cells, not only more cells express the TCR, but the cells that do express the TCR express a lot more of it. And why that's important? That's very important because the T cells with a weak TCR will not activate. So what you're seeing here is the expression of IL-2 and interferon gamma. These are two very standard activation markers for T cells against varying concentrations of their target peptide in each case. So three different TCRs being compared here. So you'll see that wild type on the left, dominant or converted to dominant on the right. In most of the cases, the non-dominant TCRs do not cause activation in the T cells, whereas the conversion to dominant, you see a marked increase in the amount of cytokine that's being secreted by the T cells. That means that these T cells have more than 1,000-fold increase in their sensitivity towards the antigen. And even in the case where a TCR does work, you're getting much better activation with a dominant TCR. And that's really the data that we initially saw that convinced us that this is very important technology in the TCR field. Because, as will often be the case, a TCR that's inserted into a T cell is going to be on the left side of the graph and not on the right, whereas these small, few amino acid substitutions will make a huge difference. I will point out that in this case, we're only talking about 3 amino acids being replaced in these TCRs. And having mapped these amino acids, they tend to be found in the border regions between the variable and the constant domain. None of them are in the CDRs, so we do not change the affinity of the TCR. We do not change the fine specificity of the TCR. All we're doing is changing the ability of the 2 chains to dimerize and form a complex with CD3, and therefore become much more effective in activating the T cell. So this is really a technology that we're going to build all of our TCR efforts on. Any of you who have an interest to have TCRs that you feel would be therapeutically useful, we're more than happy to talk with you and explore any possibilities around that area. And so this is really my last slide. This is what we're interested in. Both our platforms in terms of the NKT off the shelf as well as the dominant TCR are very broad platforms. So we're looking to work with partners to expand these platforms to new areas, new indications, new targets, both in terms of new targets or targets that you might have. And obviously, we're looking for partners to out-license some of these products and facilitate development into further stages ahead. And any of you who have technologies that you feel would benefit what we're doing at Cell Medica, of course, we want to hear from you. We want to continue developing our platforms through partnerships. So thank you very much, and please feel free to talk to me about any of this afterwards.